Welcome everybody around the world. We have people around the world for joining our webinar about how to predict drug-induced nephrotoxicity during drug discovery. My name is Errol Byron, COO at Cell4Pharma, and I will be your host for today. At Cell4Pharma, we reduce the risk of drug failure during the clinical phase of drug development by accurately predicting nephrotox at an early stage through our ciPTEC cells. The agenda for today, an introduction of the speakers, the presentation, and all of your questions can be asked afterwards during the Q&A. But without further ado, I'm excited to introduce Dr. Anna Karen Sjögren, Discovery Safety Specialist, Associate Principal Scientist and Safety Lead in Drug Discovery Projects in the Cardiovascular, Renal and Metabolism Safety area at AsraZeneca. She has a PhD in molecular medicine and over 8 years experience in delivering safety strategies in drug discovery projects, as well as hands-on experience of in vitro and in vivo experiments, both within and outside drug safety. And Dr. Michael Pearson, Director for Safety Science and Discovery at AsraZeneca. He has a PhD in medicine and neurobiology and is responsible for discovery safety in the cardiovascular, renal, and metabolic disease therapy area portfolio, from target selection to candidate delivery. He leads and manages a team of partners who design project-specific safety strategies to predict, quantitatively assess, and mitigate target and chemistry-related risks, and deliver holistically optimized clinical candidates. Dr. Anna Karen Sjögren, if you would like to share your screen, then the virtual floor is yours.
Yes, thank you for that kind introduction. And then I'll see if I can manage to share my screen. Let's see. Are you able to see the screen now? Yep. Okay. Yeah. Great.
Yes.
Yep.
Yeah.
Thanks to start with for having this opportunity to share the work that we have done on this topic. And what I will present today is how we work with predicting drug-induced nephrotoxicity in drug discovery. And I will start with describing the work we had done with the ciPTEC cells, obviously, and the high content imaging assay that we have developed. I will also touch upon some other aspects of the work we do in other and some other models as well. But without further ado, I'll go into the presentation. If I can manage to change the slide, which I didn't now when I'm in the screen mode. Okay, that was. Okay, hopefully it should work now. Can you see the next slide here now? Okay, so first of all, I will start with sharing some thoughts on how I view why we would invest in at all in developing and using new in vitro kidney models to predict drug-induced kidney toxicity. And as probably a lot of you in the audience already know, drug-induced kidney toxicity is a relatively common adverse effects on many marketed drugs still, and around 20% of acute kidney injury in the clinic is estimated to be caused by kidney toxic drugs. And we can also see that this is still an issue within drug discovery and drug development, where roughly 5 to 10% has been estimated across development programs in industry to be having a safety-related attrition that is due to kidney toxicity. And it's also true then, or has been seen in historical analysis within our company. And it's also been noted that it's quite often seen a bit more in the later stages, so to say, also pointing towards that we need better models to steer away and predict this kind of toxicity earlier on in the discovery phase. And there hasn't really been that many standardized or any, I would say, standardized in vitro screens that have been really used as standard for predicting drug-induced kidney toxicity. And talking about AsraZeneca in general, and you also mentioned in your introduction there about the therapies areas that we are working on, and that also together with that we are expanding into more new modalities as well, not only looking at small molecules, that is also contributing to an interest in and the importance of working to reduce drug-induced kidney toxicity. So now we're going to see if it works. Yeah, that works better. So this slide is to show you a bit about how, where to kind of put and where to place those kind of assays to assess drug-induced kidney toxicity. To kind of illustrate a bit on, it's really about, as I see it, use a good like fit for purpose thinking on what is the aim and what is it you want to do, because this is, I'm not going to all the details here, but this is highlighting the different parts in the drug discovery. And you see here the in vitro safety part in the earlier phases, really about then impacting the design and the selection of the molecule that has a good good safety profile, so to say. And then it's about identifying compound related factors and that you can still then influence and choose a chemistry that doesn't have this risk. When you come further down the line, it will be more about building a translation understanding if you still have risks related to the target or to your compound that's associated to kidney toxicities, and then to really develop this quantitative understanding and risk benefit of the selected molecule. So this is also like where to have this in mind when talk about what kind of assays that you would need. So if you look more historically, so to say, time ago, you would more or less have mostly to rely on in vivo findings and in vivo assays in terms of then looking at histopathology in vivo tox studies in preclinical tox species, and or also to look at biomarkers, but they are also quite often exploratory. But this is also really an emerging field as well. But this is not the topic of today about biomarkers in particular. But with this said, we were then looking on what we could do in terms of in vitro assays, and then what kind of need you would have. And to be able to say, like I said, to having impact and be used in this part in the discovery, it really has to be a quite simple, but still, of course, relevant system with a high throughput and a turnaround that is not that can generate data not too long time, and also has to be quite cost effective. And so those are different factors that we were thinking of in terms of what could be a suitable assay for us. And you could also then think about there could be other needs as well. I think then more when you go a bit further down the line again and further down the process, you would also think that you can have a need for a bit more complex system, and you can, yeah, there's opportunity to invest then in something that maybe doesn't have the same throughput and might also be more cost more, and so to say, but could still be needed for if you need a more increased physiological similarity. But again, that really depends on the question you have. There will be more, again, a fit for purpose thinking here. So with that said, I will go to this slide to see and share with you what I think personally is kind of the different keys to really having a good model in terms of kidney toxicity in vitro. And first of all, we already touched upon that, is of course the physiological relevance of the model, meaning that you could have, in this case, we talk about the ciPTEC cells, we've come back to, then being a cell line that has an improved phenotype, for example, around the transporters being expressed and functional. We can talk about even much more complex systems like different kidney-on-a-chip systems and MPS, which they are also called, and other more multicellular models such as stem cell derived kidney organoids. And I will also touch upon those two last bullet points again in the end, just to give you a bit flavor of the different kinds of models that we work with. But main topic is really about the 2D high content imaging assay in these ciPTEC cells. It's not, however, only about the kidney model or the cell type used. I think it's really as important to think about the appropriate toxicity endpoints. And I think that is really the key for this assay that I will talk about today, is to include not only like one single endpoint like cell death or something like that, to really more look at multiple endpoints and assays that also then include more kind of say subtle endpoints that would reflect more like different kind of functional or morphological changes to the kidney cells. And that is something that is also a key, I believe. And as I said on the previous slides, to apply a fit for purpose approach, meaning that you would need to match the model to the question you have and also the stage of the value chain, such as, I mean, complexity versus the turnaround time and costs and other factors. So let's go to the next one. So, and it's like I said, is the main topic of today, to show you the work that in the assay that we are using, which is the multiparametric high content screening assay in the ciPTEC OAT1 cells. And you, probably a lot of you here, but maybe not everyone, is familiar with the cells that we're talking about. So this is then the conditional proximal tubal epithelial cells. This is transfected with OAT1, so we call them ciPTEC OAT1. And I might probably refer to ciPTEC only in a couple of times, but I'm talking about those ciPTEC OAT1 throughout the discussion today. And those cells were isolated from a healthy volunteer and immortalized. And you see just a few of the reports and references here listed as well. And these cells, they are proliferating at 33 degrees, and then they differentiate when you culture them at 37. And they have a lot of different proximal tubular cell features, expression of the relevant markers. And as I also mentioned already, they have been also then shown to express different transporters such as OCT2 and also other transporters listed here. And this particular ciPTEC cell line that we used has been transfected as well then with OAT1. The assay, so what I did when we started this work was to really test out really a broad range of different dyes to stain different types or to kind of capture different things that might going on in the cells. So in the end, we ended up with 5 different fluorescent probes that we choose to really cover those range of disturbances or to cellular structure or processes that we thought or was shown to be the most useful ones. And that also then were able to be multiplexed and used together with fixation. So again, that is really to make it more handy, so to say, in everyday work when you can fixate them and don't have to do it all live. So it's what is used here is the Höchst, which is by itself very useful. I'll come back to that. So it's not only about counting the number of nuclei, but you can use this also to really look at different structures that are and morphology changes into the nuclei of the cells. We use MitoTracker to stain mitochondria. And again, that by itself then can give you a lot of information around the function and structure of mitochondria in the cells. We use TOTO3, which is staining cells that has compromised membrane integrity. Yeah, and I forgot to say here, you see the color is here also a bit. The blue is the Höchst here, and the red is staining is the mitochondria stain, the MitoTracker, and the green here is then the TOTO3 in this example. We also used phalloidin to stain actin cytoskeleton, because that was something that we thought and was kind of known before from the literature that could be a relevant and a good marker to look at cell health in proximal tubular cells. And that's the red staining in this part here. And it was just more to help on that analysis. We also included this cell mask dye, which is helping to make that analysis work in a good way. Oh, I still have some issues with changing the slides. I'm sorry for that. Okay, so yes, that's the assay, that's the cells. Then it's around what and how you would be able to find out if in a model is really predictable and how predictable it is. You need, of course, to then look at a couple of compounds, and I would say you need quite a large number of compounds to really make this really useful. We decided and ended up on a set of 62 drugs in total, and with emphasis on drugs. So we used actually what would be defined as drugs more, and did not use like things like different solvents or heavy metals and such, really focusing on the what we thought was most relevant in terms of drug discovery. We try to cover different clinical indications, and the classification was based on both the previous literature, but also clinical findings and information then in different databases. So we classify the compounds as being nephrotoxic or non-nephrotoxic to start with. Then we also then subclassified the nephrotoxic compounds into compounds that were known and had evidence of causing a more direct toxicity to proximal tubular cells, and had that as a subgroup. And the other subgroup then, obviously the 18 other ones that we call other nephrotoxic, did not have the similar direct evidence for such proximal tubular specific toxicity. Doesn't mean that it couldn't be, but it was not the main findings or information that was known about those compounds. And we dosed those compounds, and that's another thing I want to clarify here. The different compounds and how we were dosing them, we were matching these and used like different ranges of concentrations based on or related to their therapeutic Cmax. So meaning that we try to cover the area where you have the therapeutic Cmax and up to like around 50-fold, or up to, a lot of cases, it has to also be up to limitation of where it was actually not soluble at the higher concentrations. But this is something important to bear in mind, that this was kind of the approach here that we were taking to take into account what the exposures are for those different drugs in the clinic, so to say. Yeah, I will try to stay quite brief on this one because there's a lot of technical points here, but overall what I wanted to share on this is that with this high content imaging approach, we have like 62 compounds and 6 concentrations, and we had those 5 different dyes. This actually ends up in a lot of data, more than like 200 phenotypical parameters. So you have a lot of data points here. You could take approach or more manually looking to what you think is, you know, the endpoints you want to look at, but we took a bit of a different approach here, doing a more unbiased approach and starting with doing like a thematic approach to do curve fitting to generate the lowest concentrations, and also 20 and 50% activity concentrations, we call it, just because this is where you would see then a 20% or a 50% change in the parameter that we're looking into. It could be either an increase, and that's what I wanted to show you on the example here, which is platin, and an increase, for example, in the variance and nuclear intensity, but you could also have a decrease when it comes to other parameters. In this case, we saw, for example, a decrease in the actin cytoskeleton staining. So that's why we didn't call it IC, we would call it activity. We use then a machine learning approach, and like I said, today I will think it'll be outside this kind of topic to go through all the details on this, and but it was a collaboration with the chemical tox group in our company, helping us to do this approach to then use you would use like a feed forward kind of process where you select a smaller set of parameters that is required then to have an optimal model in respect to all this data and all the different measures. And in this way, we could identify the parameters that was kind of perform the best in terms of separating the non-nephrotoxic and the nephrotoxic compounds from each other. And we then ending up with different parameters related to mitochondrial health and nuclear morphology, actin cytoskeleton, and membrane integrity. And this is a busy slide, I really know that. And this is from the publication, and you see also the link to the publication that we have on this topic. And this is to show you the sensitivity and also the specificity, or like the false positive rates for the different individual parameters. And we have showed this in a way that we have again then put it into relation to the Cmax of the compounds to kind of show you that you can look at the different cutoffs here, and then you can see what is the predictivity, what sensitivity do you have, and what specificity do you have. And if you look at those individual parameters, they are ranging a bit, but you still have maybe around, yeah, sorry, I should also say that the red one here is all the compounds, and the more kind of orange one is the proximal tubular toxic compounds by itself. And this way we could kind of use this to then set a threshold, which we call like a TI there, and then you could say whether they are toxic or non-toxic if they are then above or a certain threshold. And I would also say that this is a point that I want to make here, is to see that maybe you wouldn't think that it's such an impressive number by itself if you look at those individual parameters. And I think the really kind of beauty with this assay is that when we then looked at all those combined, meaning that you would classify something as being positive, so to say, in the assay as having a hit in one of these parameters, then you would have a much higher sensitivity, but still keep the specificity. And that is what I would show you with this one. So we were looking at as what is the best combination, so to say, of parameters. And we're ending up with actually limited down to like 4 parameters. With using those 4 parameters, that was around membrane integrity loss and mitochondrial and intensity of the cytoskeleton, and also nuclear size. For each of these drugs here, we have then used the lowest TI, meaning the ratio between the concentration where you saw a response and the Cmax, and in any of those 4 parameters. And then we plotted this against the different sensitivity and false positive rates. I understand it's not probably the easiest to follow all the details on those kind of calculations, but you can then see here that the sensitivity and the false positive rates in this figure. And here you have then plotted against the lowest TI. And if you then take, which I hope will work now then, yeah, a threshold, so to say, you can more or less put that wherever, but here, I mean, you can illustrate that with where I put this threshold then of a 200, then at this point you actually if you have a compound called nephrotoxic, so to say, if you're below this, we have a sensitivity of 75% if you look at then only the proximal tubular toxic compounds, and with still keeping actually a specificity of 100% in this case. Of course, this is the validation data that we have looked at, and of course, you can need a more, probably an even larger kind of set than compounds to kind of test if that is that specificity can still be kept that high. But I think that is really key for us also, that we don't want a model that is, how to say, is equally important, I would say, to have a model that is having good specificity, so that we wouldn't kind of throw out compounds for the wrong reason, so to say, too early on, if you would have something that is not specific enough. So that's why we had that put as a quite a high factor within this whole work, that we wanted to think about the specificity as a very important factor, and also the reason for taking quite a lot of non-nephrotoxic compounds into the validation as well, to make sure we really assess that part and not only the sensitivity part. So I don't know, yeah, we'll probably come to question in the end, we say, right? So therefore I move on actually to come back to this slide, where I wanted to then highlight that we thought that this multi-parametric high content assay in the ciPTEC 01 would actually then be an assay that could enable this kind of this screening to be used quite early on, so to say, to be able to mitigate drug-induced kidney toxicity in our company. And we are using this model, and I would say right now we are using it as a, how to say, ad hoc, in terms of meaning that we are applying it to projects, and we have some examples where we have had a good impact on being able to show differences between different compounds within a chemical series and such, and to be able that have helped project to go with compounds that seem to have less effects on those parameters that we are looking at in this assay. So with that said, I wouldn't want to go into, just give you a bit of an overview of the other types of in vitro kin assays that we are also working with. And for those, we are particularly not right now using ciPTEC cells, but that doesn't mean at all that those couldn't be kind of used for that as well. So this is something I want to share more as a general thing in terms of more complex systems that we are also looking into, where we would potentially see that we need for a particular question some feature that would not be kind of wouldn't be covered with this more higher throughput kind of system as the ciPTEC. So that is what I will show you next. But I actually realized I wanted to say one more thing that I don't have a slide on, but that work that we have done also that really shows the usefulness of this multi-parametric high content assay in the ciPTEC cells. We did run those same validation compounds to one of our more general cytotoxicity assays, so to say, like our standard in vitro cytotoxicity assay, in a completely not any kidney cells, really, like I said, a general cell type, to see how much kind of value this assay with the ciPTEC cells is adding to that. So we just didn't only, pick up compounds that are cytotoxic in general, but that was really clear with that analysis that we did pick up. I think it was around, there was like at least more than 10 of those direct proximal tubular toxic compounds we only picked up using this multiparametric approach in the ciPTEC cells that we would have missed if we would only have run them in the general cytotox assay. And I think that's really, really an important part here, and that shows that there is a value, and especially for compounds that are having some kind of mechanism that it makes it more specifically toxic to, in this case, proximal tubular cells. And among those that we picked up only in the ciPTEC OAT1 cells was also then including those compounds that are known to be taken up, for example, actively by OAT1. So that makes fully sense that these are not readily detected in the normal cytotox assay. Yeah, so with that said, I wanted to show you some of the other work, as I said, that we are looking into in a more exploratory way to more mechanistical studies and risk assessments within our company. And this is a kidney proximal tubular MPS system that we are working with. The actual chip is from Nortis, and we actually, in this case, we have used cryopreserved renal proximal tubular epithelial cells in this, and you can then place those cells into those tubes, and you will then have like a proximal tube in here, and then you have also the flow of media. And we are with this seeing, and this where I try to have a movie, but let's see if it works. No, I can't show that. Okay, I'll go to this anyway instead then. So this is staining that shows that they have tight junctions expressed in those cells, and we have also looked at gene expression, and this is work from a postdoc that are working with us, Tom Niskens, and this is really just to give you a flavor of where I see that this type of model we could see as having a use, for example, if you want to do more long-term or repeated drug-induced kidney toxicities, because you can have the cells in this kind of system for a longer time. And again, the cell type can probably be kind of different types of cells can be used in this kind of system. And we have recent published work using this system, and that's linked down here on that. And we actually, just to give you, if you're interested in learning more about ASO-induced kidney toxicity, we also just got another publication accepted this week. So that you can always have a look for that if you have a particular interest in that. And the last piece in this whole puzzle I want to mention is that we also have access in our company to iPS-derived kidney organoids. And this, I would say, is not, as I see it at least, as a kind of a way forward in terms of screening a lot of compounds, but more to enable studies of particular genes or proteins that you might want to use, that where you maybe need to have like the other cell types involved for that functional response that you're looking at too. So again, that this was more to show you that I think it's probably not like one single system or out there that would really answer all the questions that you would have in this area. So that is the point I want to make with that. So again, I think this will be more for mechanistic studies, so maybe complement in vivo studies for the risk assessment when you go forward. So I think the take-home message that I wanted to give you before I open up for question is to say that both the culture model and the cells is very important, but also the endpoints that you are using. And yeah, there's probably, at least not so far, any completely perfect model out there that is suited for every kind of question you would have, but you have to think about what is your needs. And in our hands, this combination of multiple cellular health parameters that was assessed then in the ciPTEC OAT1 cells was really useful in terms of an assay to predict and identify, and we have examples where we've been able to use it to mitigate drug-induced kidney toxicity in our early projects. But there could still be need and for more complex models for more specific questions in terms of more investigations into different types of mechanisms that might be not covered in a simpler system. Yeah, and by that, I think that's what I wanted to share. And of course, there's a lot of people that have been involved both in this company in AsraZeneca, but also this all started out as a collaboration with the Radboud University around using those cells. And with that, thanks again, Errol, for the invite and to be able to present today, and open for questions then.
Yeah, thank you, Anna Karen. Yeah, there is already one question.
Yeah.
From Amrit. I see that Michael, you answered it already via the chat, but it went to only the panelists.
Yeah, but I think that let's have Anna-Karin answering the question anyway.
Let's see if I can see the questions here then. Can I see that on
I can read it for you otherwise.
Yeah,
so
please do.
The question is, when it came to the validation compounds, whether there is a difference between parent and metabolites being nephrotoxic, because they are wondering whether the system can pick up toxicity from both parent and metabolites.
Yeah, whether if those cells are metabolic, I say, competent or so, I guess, I mean, we haven't really looked into that in any detail, I would say, so that could answer that if that would always be the case. I know there are some, features of those cells that were pointing to that there is kind of metabolism in those. So I would say partly yes, but also that if you would look specifically for a metabolite, you could of course also test that specific metabolite, and that's something we have actually done in one of our projects actually, where we had a question around if a potential metabolite was causing a kidney toxicity. We more took that approach of actually running the actual metabolite in the assay. I don't know if that answered the questions, or if you want to add something there.
Well, I think it's fair to say that when we developed the assay, we didn't want to divide compounds into whether they were mediated by a metabolite or the parent compound, because that would sort of more mimic the situation we have with our more unknown compounds during drug discovery. I think that the cells have some limited metabolic capacity, but I think in many cases it's mainly the parent toxicities that we're picking up, unless we actually expose them to the metabolite, as Anna-Karin said.
Okay, thank you. Are there any other questions? You can ask them in the chat or in the Q&A section. I'm just waiting for some questions to come in. Wait.
Let's see if I can manage.
Amrit is replying. Very informative overview. Thank you for the response. One more question. Did the ciPTEC show a difference between molecules that cause something less injurious, like, wow, that's a, can you read that, Michael?
Yes, phospholipidosis.
I can see it now as well. VS clear degeneration. I guess if that is meaning, if we see, I mean, a difference on I mean, of course, I would say there is definitely a range in terms of, how toxic the different compounds were shown to be, so to say, and of course some of the compounds in the validation assay is, you know, showed toxicity as quite, you know, dramatic, while others were more showing more, like I said, subtle changes, so to say, which I think is one of the keys here as well. So I would say yes, you can see those differences, but I think the good thing also is that you would be able to pick up more of those toxicities that are not maybe where the readout doesn't necessarily is just like the loss of cells, but you would rather see some changes to the morphology or something, at least at the lower concentrations, you get a bit higher sensitivity. But with that said, I mean, specifically that type of mechanism in terms of, for example, phospholipidosis, I mean, I don't know. We actually did use also stain to stain at lysosomes as well, because I know there are some, you know, links to that as well. It was not so particularly useful in terms of in general for predictions, but that could maybe also be because of technical limitations as well.
Yeah, I guess it's worth saying there that while it didn't really add much to our prediction model, we can sometimes use phospholipidosis like stains in the cells in order to address a certain question. Yeah,
exactly. So that is probably definitely a possibility, as you say, to do that when you have more, for example, maybe you have some concern on for that particular toxicity based on the structure of the compound or so. I mean, you could definitely expand this toolbox, so to say, of different dyes to also include other dyes, definitely. I mean,
I guess a very easy answer is that we tend to use this assay very pragmatically. So we do one version of it for just routine screening of compounds, where we are just using the validation set. But then, of course, when we have specific questions, we also tailor the assay accordingly to that particular purpose.
Yeah, and that could, for example, as you say, Mika, then be including some additional markers or dyes or so to complement.
All right, we have another question. Would the process be similar when testing biologics?
Yeah, we haven't, and that's a very good question, an interesting one. I mean, so far, I mean, this validation was obviously then done with small molecules. That was our kind of main, at that point, I guess, main interest, but also like the starting point, I would say. I think this is something that is probably doable, so to say, and there's something that we have started to touch upon, where we haven't really done any kind of validation there, so to say, but at least, yeah, very limited so far on what we have done there. But I think in general, it should be able to apply this. I guess it could be around the accessibility into the cells and questions like that, of course, that you have to understand that part. But
all right. Are there any other? Oh, wait, another one. Jean-Marie, any experience with predicting drug-induced AKI to CKD transition in your in vitro models?
If you would know, I wouldn't say that we have looked into detail on that kind of particular, how to say, compound that would be, if that is the question, that has been kind of causing AKI, and whether that is then translating to CKD. No, we haven't really looked into that. We are, I mean, in the MPS system, working on a chip system that I also touched upon here. I think, you know, that is a model where you could potentially look at more like disease type of approaches to kind of more mimic like the disease, but in the high content in imaging, as you could probably do that as well. I mean, we have some, definitely would be an interest in looking more disease type of models, if that's part of that question, but we haven't really done any kind of proper validation in sense. It's more like exploratory work.
All right, thank you. Another one. Have you looked at the ciPTEC 083?
The ciPTEC 083, we had some access, if I remember correctly. Now it was starting to be some time ago now. We did some work on that, but at that moment I think they were, we are for different reasons chose to look at specifically the ciPTEC 081 because we thought that was the more kind of broader applicability at that point in time for us. So no, we haven't done a similar evaluation with those.
All right, thank you. Any other questions? I'm checking the chat or the Q&A section. I don't think anything is coming in. We got a comment from Amrit. Thank you for the response. Great presentation. A comment, it would be great to see if there were cases where one in vitro model was more relevant than others in future presentations. Great job.
And that is a very good overall comment, I think, because that is relating a lot to what we do in the drug discovery phase, because the more complex models that Anna-Karin was talking about, like the kidney organoids and the Nortis system, the throughput there is of course not very good for us. So we only apply that to more specific cases. They might be more relevant, but using the ciPTEC cell assay as a general filter to get rid of the worst compounds is usually well worth our time. So it's about the cost, but it's also, as Anna-Karin said earlier in her presentation, about the turnaround time, because if we want our chemists, for example, on a small molecule project to be able to react on the data that they provide us, so that they actually can make molecules that don't suffer the same liabilities, it needs to go down to around 2 weeks turnaround time. And we need to be able to test a lot of different compounds to generate SAR. And we will basically never be able to do that in the more complex systems.
No, I agree, Mikael, that's a good comment, as you say. And it's also part of why I wanted to mention that kind of comparison we did also internally with our other kind of assays, because just that will be kind of the obvious thing, as you said, Mikael. Is it worth than the kind of investment in terms of how many more compounds would a particular model pick up versus a simpler one, which is actually the comment here is also reflecting just that. So that's why I think that this kind of stepwise approach, as you said, Mikael, where you'd go down filtering a bit more and more is a pragmatic approach that we are using, so to say.
All right, thank you. We have another question. Have you tried disassociated HIPS kidney cells in similar assay?
So the one, what can you repeat that question?
Well, basically the question is whether we've used human-induced pluripotent stem cells, but dissociated in a similar assay.
No, we haven't. Is the simple answer to that. If I guess that's a limitation on how much work we have been able to do now.
All right.
Yeah, I think that it's not like we haven't thought about it, but I think one of the realities is that it takes quite a lot of validation work, and once you've sort of settled on a system that works as a fit for purpose model, you tend to stick with that, because otherwise you're spending a lot of your resources in trying to develop or testing things where the increased benefit might be a little bit too small in comparison to the resources required.
Yeah.
All right. Thank you. I've just in case I have put my email address in the chat. If anybody wants to ask Anna Karen or Michael anything, they can always send me an email or Martijn for that matter. They can always send me an email about anything related to ciPTEC, and I'm happy to answer all of your questions. Are there any other questions? If not, then we'll end it here. I think there are no more questions. So thank you very much, Anna Karen and Michael for your time and the presentation. Thank you very much. And if there are any other questions from people coming my way, then I'll definitely let you know. But thank you very much, and I hope to speak to you soon.
Yeah, thank you, and thank you everyone that joined for your interest. And yeah, please reach out if there's more questions, as Harold said.